So, hi, I'm Barbara from Malibu Divers. For those of you who don't know me, happy to help you with anything that's going on. Tonight's speaker is Dr. James Bella. He's coming in from New Zealand. He's Professor of Marine Biology, Marine Ecologist at Victoria University of Wellington. I read an article about sea squirts, and lo and behold, it actually is more than just sea squirts. You know, James specializes in sponge ecology and temperate Mesophotic reefs. I think that's how you say it. Leading deep sea research with ROVs. His talks covered deep sea exploration, marine biodiversity, ROV science, and ocean conservation. And we hope to see some of that underwater footage. So recently, I just saw on your Instagram site that you just won an award from the Royal Society of New Zealand for Distinguished Research Fellowship, one of only 2 awarded per year. And correct me if I'm wrong, James, it looks like you're always recruiting for new people.
I am, yeah. We're always looking for new PhD students in particular, and master students, yeah.
Cool. So thank you for being here, and the floor is yours.
Right, thanks. I'll just share my my screen, and then I'll get rid of that, and I will show. There we go. Can everybody still see my screen?
Yep, I can see it.
Cool. Right, thank you very much, Barbara, for inviting me. I can't see anybody else go. Thanks very much for inviting me. It's a pleasure to be here and chat to you, Barbara. And I did have, I guess, some issues with the time zone difference, and which I'm really glad that we were able to resolve, and I was able to come and chat to you. I guess as well, before Barbara contacted me, I had no idea what the environment was even like in Malibu, and I'm glad I do a bit more now. What I'm going to do, I guess, today is tell you a little bit about some of the research that my group does. I'll maybe talk for, I don't know, 25, 30 minutes. Quite a bit of it is video. And then hopefully it shows up really well on your screens. But tell you a little bit about some of the places we visited and explored, and tell you a little bit about the science and some of the reasons by it. And I guess some of the threats and impacts on the types of ecosystems that I work on. So I guess I should start by acknowledging the Royal Society that funds a lot of this work, and there's a whole bunch of other people at the end who have contributed to the things that we do. And also I kind of talk about it as we and me, but a lot of this work is supported by a whole bunch of other people as well, particularly lots of students who contribute to the research that I do. Okay, so I guess I'll start a little bit about me. So I'm a professor of marine biology at Victoria University of Wellington in New Zealand. So I don't know if any of you have been lucky enough to visit New Zealand, but we're at the kind of bottom of the North Island. We're in what's called windy Wellington, where the weather is pretty much always horrible, and very, very windy, and very difficult to dive. So it means I do quite a lot of my diving in other places in the world, and I'm going to talk briefly about some of that. So I'm not a born and bred Kiwi. I was, first of all, I'm actually from the UK originally, and I did my PhD, so my degree in Bangor University in the north of Wales, and then I moved to Ireland, where I spent 3 years doing my PhD. And then after my PhD, I traveled around, did a few jobs in the UK, and then I moved to New Zealand in 2006. And that's where I've kind of been ever since. And that's where a big chunk of my my research was focused. So I guess Ireland is probably at the cooler end. The warmest temperatures in Ireland are kind of at the warmest temperatures I think you kind of get in Malibu. It's kind of maybe I guess summer temperature would be 14 or 15, and it gets down to about 5 or 6 degrees, you know, in some of the coolest parts and some of the inland places. So it's a cold water diving. So after I'd done my PhD, I very much wanted to start doing tropical diving. So I started a research program out in the Wakatobi, and this is a picture from the Wakatobi. One of the cool features for me is that it has incredibly high sponge diversity, and I'm going to come back to sponges a bit later on in my talk, not in relation to the Wakatobi, but more generally. And it's not uncommon to see these sponges that we think have been sitting there for, you know, anything up to maybe 1,000 years. Just sucking in water and spitting it out again. I'll come back to that later. I wouldn't describe myself as a particularly well qualified diver. More of a very experienced diver. I have a bunch of qualifications, and a scientific diver. I've done maybe, I don't know, 3,000 or 4,000 dives. Most of those, I guess, apart from my early dives, are science dives. Dive pretty much all over the world. This is a picture taken of me not that long ago in Fiordland, which is a quite unusual place in New Zealand, and I'll probably come back to this place a little bit later. And what I'm kind of sat next to or floating next to there is a giant black coral, which is one of the things that my group works on. These big black coral structures can be anything up to 300 or 400 years old, and you can see as they get old, like this one, they get encrusted with all sorts of cool beasts like sponges and ascidians. So what I want to talk to you about is I guess a zone in the ocean and a bunch of ecosystems that most people aren't particularly familiar with. And the main reason for that is because they're ecosystems that are not particularly difficult, not particularly easy to access. But in many cases, for us as divers, they're actually not too far away from where we reach in the oceans. So the mesophotic zone, or mesophotic means middle light. So it's typically defined as the area that exists, or the ecosystems that exist just below the kind of kelp zone. So typically in temperate waters, that can be anywhere between about 25 meters, or if the light penetration is really low, it can be shallower than that. And that picture I showed you in Fiordland earlier, those are mesophotic ecosystems, and they occur in much shallower water because of the unusual environmental conditions there. So these mesophotic ecosystems are characterized by still having some light, but very, very low amounts of light. So there are often things like encrusting algae, the kind of calcareous algae that's kind of pinky in colored, covering the surfaces. But these animals are largely dominated by animals. Okay, so we're talking about animal-dominated environments rather than shallower kind of kelp and seaweed-dominated environments. Now, the mesophotic zone itself has only really been recognized as a kind of distinct thing in kind of recent times. People just used to focus on the deep sea. They focused on the shallower environments that scuba divers could get to, and this zone of the ocean had been pretty much ignored. How do we get to these environments? So we use a bunch of ROVs, so remotely operated vehicles. These are different to the kind of ROVs that you might see with NOAA, where they've got a huge, massive, great boat, and it costs millions and millions of dollars. And you need a team of 30 people and technicians and engineers to operate it. These are what we call kind of small mini ROVs, and this was our first small mini ROV, which is this is probably one of the first ones that became kind of available on the market and actually produced relatively high quality images. It only takes a couple of people to deploy it, relatively low cost, maybe 30,000 American dollars to buy one. Has a grabber so you can catch samples. And this was our first I guess, exploration of these mesophotic reefs with this little mini ROV. We've since moved on, and one of the reasons I'm actually sitting in my my kitchen as opposed to in my normal office is I have our ROV next to me here. I'll give you a proper tour. Maybe you can hopefully see it there. I move the thing around. You can still see me. I'll give you a bit more of a better view a bit later on. As you can imagine, my wife is delighted that I have our ROV sat on our kitchen table. But this one is quite a bit more expensive. It's probably about maybe $200,000 to purchase, and requires quite a lot of maintenance to make sure it operates correctly. It has a depth range down to about 250 meters. But if we replace the dome here with a different type of dome, it can extend down to about 800 or 900 meters. So it has the potential to reach really quite deep environments. But we're typically only exploring areas down to about 150 meters because that's the limit of light and the limit of the mesophotic zone. So that's where most of our studies are focused. This ROV is super cool because we can take it pretty much anywhere we want to. It packs down into one case it's 32 kgs, one case that's 20 odd kgs, and then another one which is just the cable, which is maybe 12 kgs, which means that we can basically fly it all over the place. We can fly it around New Zealand and deploy it wherever we want. Also, we only need a couple of people to deploy it and one person to generally pilot it. The other really cool thing about this particular ROV, which is actually made in New Zealand, is that it's kind of the ROV is built, if you like, around the camera. So it has a Sony A1 located inside it, so a very high-end camera, which means it can capture really high quality video at 4K or 8K or 50 megapixel pictures. So some of the discoveries, what I want to do is hopefully these videos look as cool on your screen as they do on mine, but I want to kind of walk you through some of the discoveries and places and things that we found in New Zealand. If you want to add places to your list of places that you should visit and dive, this is some video from about 60 meters at a place called the Pool Knights Marine Reserve, which is up at the top of New Zealand in a place called Northland. It was recognised by Jacques Cousteau as one of his 5 most favourite places to dive in the world. It is a truly amazing site. It's a bunch of islands in the marine reserve that's kind of out off the coast of New Zealand, so it has a kind of big oceanic influence. Not much sediment, and you get these amazing encrusting communities that actually extend from about 20 meters down to hundreds of meters. So all of these animals, you can see this is the Mesophotic zone. You can still see it's quite light still, and there are lots of encrusting beasts. You can see it in the corner there. You can see the light upwards. So there is still light down in these environments, and that's kind of what characterizes the Mesophotic. If we have a look at some other environments, this is heading just slightly further south. You can see that the white kind of tree-like structures in this case are what we call black corals. They're kind of a distant relative to normal corals that you'd be familiar with. And you can see these environments are kind of teeming with fish and life. So their Mesophotic ecosystems tend to be really diverse, and they also tend to be really different to what you find in the shallower waters. And black corals are quite common in these environments. In New Zealand, black corals are a protected species. They're very delicate. They're also very long-lived, so they don't recruit back to these environments very often. We've also in the Mesophotic zone found, I guess, actual kind of coral reefs. These are stony corals. These are hard corals at a depth of about 105, 110 meters. This is again a little bit further south in New Zealand. These are again slow growing. They're protected, particularly delicate to things like trawling and fishing activities. You imagine, if a fishing line gets wrapped around that, it's going to snap off chunks relatively easily. We've also found things like this. These are the ones that excite me most because I work a lot on sponges. These are glass sponge beds. Glass sponges are a very ancient type of sponge. Again, organisms that are thought to live for really long periods of time. Nobody knows exactly how long, but it's likely to be hundreds of years. And again, you see that these environments are super dominated by fish. What you're seeing there in the picture is a type of fish that is highly prized in New Zealand. It's called a kingfish. Great if you're a fisherman for fighting and catching, and exceptionally good eating. They're what all the recreational fishermen try and target in New Zealand. But you can see these environments are teeming with life. There's no algae anymore. They're all completely animal dominated. But if you look really close, you can see there's a little patch of pink down here. That is a type of algae, an encrusting calcareous algae, and that can survive because there is still some light in these environments. If we skip down to a bit closer, nearer to Wellington where I am, these are, again, you can see as we're moving kind of down further south, down through New Zealand, that the communities look quite different. So there's a lot of variation in the types of organisms that we find and the different species that we find as we move from one end of New Zealand to another. They're not all the same. I guess that's maybe what you'd expect. New Zealand's a big place, but we found I don't know, probably 50 or 60 different types of communities so far that are distinctly different to each other in the different places that we've visited. And again, you can see a bunch of fish, but different species this time. This is taken from Fjordland, and this is taken from about 40 odd meters. That was the place where the black coral was in the first picture I showed you. These again, we know very little about the ecology of these. They're called a red coral. They're actually not a coral at all. They're actually a type of hydrozoan. Again, similar relative to corals, but super delicate, and we know very little about their ecology, but likely to be really impacted by things like fishing or ropes and things from pots and those sorts of things. Probably though my most exciting find, I think, across New Zealand, and this is probably what Barbara saw and contacted me about because there was quite a bit of media about this, was some work that we did at the very bottom end of New Zealand. So pretty much all of those videos that I showed you before, one of the most dominant things that we find is sponges in these environments. However, at this place at the very bottom of New Zealand, the thing that we came across were these gardens of sea squirts, so ascidians, that literally covered the floor. We haven't seen anything like this anywhere else in New Zealand, And to my knowledge, in these types of environments across the world, nobody else has seen anything quite like this. So there are some sponges in there, but pretty much all those weird shapes, particularly those egg shapes, are all different types of sea squirt and ascidians. This is down at about 110 meters. You can see the ROV, we've got lasers as well, so we can size these different things. And you can see that basically there isn't a bit of bare space. The entire seafloor is covered in these organisms. And you can imagine, again, any fishing activity, whether it's dropping pots or trawl lines or even recreational fishers are likely to damage these environments. These ones are particularly cool because the white sea squirt here is a new species. Nobody had described that before, and nobody knew what it was. So it was quite a cool find. The slightly sad thing in New Zealand, and this is no different to other places in the world, is there's little recognition still of these types of environments, which means that they're not included in many of our marine reserves. So our marine reserves have tended to focus on more shallow environments where people have seen cool things when they're diving, and those are the things that we've protected, and we still have most of these environments that are not protected. So I guess a little bit of sciencey stuff and a little bit of related to what I'm particularly interested in. So I kind of, Barbara alluded to this to start with, that one of the things that my group works on is sponges. And hopefully you can see from those videos one of the reasons why I might work on sponges, and that's because they're really dominant and really important in these mesophotic ecosystems. But for those of you who aren't super familiar with sponges, they are one of the most ancient organisms on the planet. They evolved around 550 million years ago. That's when their first sponges were seen in the fossil record, and they've pretty much not changed very much over that time. They're really simple organisms. Their body plan is based around an architecture of water canals, where they have specialized cells that pump water into the sponge's body, strip out all the tiny particles, and then spit the water out again. Basically, if you're unlucky enough as a tiny particle to be sucked into a sponge, the chances of you leaving are really slim, as they have kind of up to 90 to 95% retention efficiency of the tiny particles. Just to demonstrate kind of how important these sponges are in these different mesophotic ecosystems across New Zealand, here are two pictures of spongy-dominated environments. The places we've been, this little graph here shows a number of different locations, and it shows the percentage of the seafloor at about 80 meters that is covered in sponges. And you can see at pretty much all the locations we visited, more than half of the seafloor is covered in these sponges. And that's, one of the reasons why I'm super interested in sponges. Just to give you a view of what that pumping looks like, and hopefully this one shows up. This is a piece of sponge that's probably about half the size of your fist. What we've done is we've put some fluorescent dye in the water next to the sponge to visualize the pumping activity through the sponge. And you can see how quickly it circulates that water. Probably a sponge pumps its own volume of water, its own body volume, of water in probably 10 to 20 seconds. So it's super efficient. So when you're swimming along the reef and you see lots of sponges, particularly in the tropics, when you've got, like in this picture here, large barrel sponges, they are basically recirculating and recycling that water at a very rapid rate, and having a really big impact on the overlying water column on reefs. So what impact those sponges have is one of the things that my research group focuses on. We've done a lot of work trying to figure out what sponges eat. They eat all of the tiny plankton in the water columns. They eat little tiny bacteria. And I'm not going to go through this in a huge amount of detail, but just to describe that we've spent a lot of time trying to work out if that feeding activity of sponges is actually important or not. We've done a whole bunch of experiments where we've measured how much food sponges consume, what food is available to them, how much water they pump, and what impact that has on the food availability that's in the water column. This is some data from one of those studies at a place called Fjordland, which I've mentioned a couple of times before. As a diving location as well, I would add that to your list. Fjordland is very exclusive and very difficult to get to. There are a number of dive charter boats that operate there, but it is amazing environments. It's a kind of dark green environment that has animals all over the cliff walls that are super cool. But one of those animals that's super important and abundant are sponges. And what we did is we measured the abundance of sponges in a number of different locations in the fjords, these different colors here, for inner parts, middle parts, and outer parts of these big fjord environments. We measured and worked out roughly how much water sponges were pumping, and we worked out how much of the available food or the plankton and the bacteria that's floating around is consumed by sponges. If we just look for the information at the outer part of the fjord, so this area here, that's shown in orange, which is where sponges are most abundant, They pump anywhere in the region of 1.5 meters cubed of water per minute. They strip out maybe up to 10 to 20,000 kilograms of carbon per day, and consume somewhere between 2 and 10% of the available carbon in the water column every day. So they're basically having this huge impact on the overlying water column in these fjord environments. And it's worth noting the scale here. These aren't small fjords. These are, fjords that would take a couple of hours to go from one end to the other in a boat. So we've had some funding in the last couple of years to study these impacts, because until we used to think that sponges taking the food from the water was kind of like the end of the story. They made the sponges grow and they got bigger and bigger. But we've had a bunch of money to try and study if that's the end of the kind of food web pathway. And what we're finding is actually it's not. So when sponges feed and they eat all of this bacteria and all these things that are floating around in the water column, the thing that they do is produce what's called detritus, or you could consider it as a kind of sponge poo. That sponge poo then gets released from the sponge and gets eaten by all of the kind of tiny little crustaceans and hermit crabs and brittle stars that are crawling around on the seafloor. They then feed on that detritus, they grow, and then the fish species, the things that we're interested in from commercial perspectives and recreational perspectives, then feed on all those little tiny critters. So sponges are really central to this idea of a kind of trophic relationship and these trophic cycles between the water column and the seafloor, and then back to the water column. The other cool thing about sponges, and this little diagram at the bottom is a kind of diagrammatic representation of what's going on in a sponge if you chop it open. There are all these different critters that are associated with sponges, from nudibranchs crawling across the top, eating the sponge, to all of these things living inside the sponge, from copepods, amphipods, little snails, mussels, that are all feeding on that detritus. And if they leave the sponge, or if things eat the sponge, then they get transferred to things that are a bit higher up the food chain. I don't know how well this is coming across on Zoom in terms of what you can see in your screen, but this sponge down here, you might see all these little tiny speckles in the kind of aquarium surrounding the sponge. These are all this detritus, this sponge poo that's being produced, and they produce quite large quantities. They can produce several grams a day, which is then fed on by all of these other organisms. The other thing we're super interested in is the potential for these, not just the sponges, but the other things to produce habitat or provide habitat for lots of different species. This little diagram shows that quite nicely here. There are lots of fish species that go down, they feed in those mesophotic environments. They also then shelter from their predators in those black corals and in between all of the sponges. This is just a little video of some baited underwater video that we did, where we've got, you can see the Mesophotic reef in the background there, and there are all these fish species that basically come along to the bait. So the banging noise is the fish in different parts trying to munch on the bait that's inside this little pot. You've got these amazing fish assemblages that are associated. Some are down there feeding, some are hiding from their predators. You can see there's a bunch of different, bunch of pretty species. This guy coming in here is quite a small one. Those are a snapper, again, a highly prized fish in New Zealand for recreational fishers and also for commercial fisheries. And they are frequently found in these mesophotic reefs, and probably go there to feed. The other thing, or the way that we're kind of approaching this idea of whether or not the seafloor provides habitat complexity, is using a technique that's called photogrammetry. You might not realize this, but what you're looking at here, this looks like a picture of the seafloor that I showed you of the video before. This isn't actually a picture of the seafloor. This is actually a screen grab of a three-dimensional model that we have made of the seafloor. So we can use a process or a technique called photogrammetry, where we basically take thousands of pictures of something and we recreate a 3D model of that environment. And that enables us to measure things like how complex these different environments are. We can use various methods and statistics to estimate how much rugosity there is, sort of how much up and downness, how much three-dimensionality there is to the environment, and compare how that changes with things like fishing effort or protective status. Just to demonstrate that the photogrammetry we've been doing as well has been at a number of different scales. Hopefully this will play. This is some work by one of my PhD students, looking at a different level. So this little sponge here is about the same size as half of your little finger. And we've taken basically 1,000 pictures of this sponge stitched them all together to create a perfect representation of this little sponge. So we're using these photogrammetry techniques to look at the habitat provided by these different habitats for things of all different sizes. So things like little triple fins or little tiny gobies will live in those spaces between those sponges, and they're all the food for all of the other things. So those are the two main areas of our mesophotic research. We're also interested in, more basic ecological stuff, why are they different in different places, what species are found there. But we're really interested in those contributions and importance to fisheries. Because if we can demonstrate that they're important to fisheries, then typically there's more interest then to protect them. At least that's a field in New Zealand. If you can show that something's important, then people perhaps put more value on them. So I guess the question is always asked is, are these ecosystems threatened? And the answer to that is we probably don't know, because they've been studied so poorly. There are lots of examples in our ROV videos. You can see here, sorry, it wasn't particularly taken for this purpose, but you can see over here there is some old ropes. These will either be from our old anchor line or they'll be from potting activities. You can imagine most of the areas we visit are fished by cray fishermen, which dump pots in these environments. And you could see from all the videos and all the pictures I've shown you, those areas would be, really easily damaged by the dropping of pots on them. But at the moment, we don't really have a handle on whether or not that is really negatively impacting these environments or not. It would seem that it would be, but it's kind of hard to know. The one thing we do know that is impacting these environments is marine heat waves. I guess marine heat waves are an impact across the world these days. They were, until probably, 5 or 10 years ago, most people talked about marine heat wave impacts on coral reefs, and that's where most people were interested because all the corals were dying because they were bleaching, and people didn't worry so much about temperate regions and cooler regions. But that's changed dramatically in the last 5 years. In 2022, 23, New Zealand experienced its worst marine heat waves it had ever experienced, and one of the groups of organisms that were most impacted were the sponges. This was some work that we did looking at one species that bleached, then subsequently went necrotic and died, and we estimated that something like 50 or 60 million sponges died. So that's million sponges died as a result of the marine heat wave impact. So these warmer temperature events can have quite dramatic impacts. There's a lot of information on this slide, but I just kind of wanted to demonstrate, how severe the impacts were in some parts of New Zealand. And this graph at the top here, the kind of dotted line shows the average sea surface temperature, averaged over the last 30 years through time, which is on the x-axis here. And this kind of more jaggedy line shows the actual temperature during 2022, 2023. And you can see that it deviates quite substantially here from the normal, and it was actually nearly 4 degrees warmer than what it would be towards the end of summer. And this caused sponges to basically bleach and melt away in these mesophotic habitats, which was very sad to see, I have to say. Yeah, so this is kind of what the view looked like. So we did surveys. This is Fiordland again, the region I mentioned a few times. This is what it looked like down the cliff. These are all the sponges that bleach white, and then they all basically went kind of necrotic and in most cases died. Some of them did recover, but in most cases the loss of colour then resulted in them all dying. I don't want to leave it on a super negative note. You saw from all of my videos that those kind of mesophotic ecosystems in New Zealand and other places where we visited tend to be in much better condition than the communities that you find in the shallow waters. And maybe that's partly to do with heat waves. Maybe they're a bit more removed from human impacts. But obviously also we're only observing a lot of these things for the first time now, and they could have been very heavily changed in the last 50 years or 100 years from human activities, but we just don't know. There are so many research questions around these ecosystems in terms of the species they support, how they work, how things recruit to them, and I guess we're just starting to understand some of those things. So that's where I'm going to finish my talk. So hopefully I've left a big chunk of time for people to ask questions. I'm happy to share anything else. I would, I guess, before I would show you, as I say, apologies, I think about a really great way of doing this, but I did want to show you our ROV. This is George, who is named after the person who provided us the money to buy the ROV. So you can see, hopefully on the front, it has a grabber just down there, which we can use for collecting samples. It's got 8 thrusters, so it's fully vectored. Also, you might notice on the top, it has a little, it's not easy to do this. Just on the top there, it has what's called a USBL, which means that we can accurately locate the ROV when it's underwater, which means that we can potentially go back to exactly the same spot. I'll bring you round. Excuse my house. Did tidy up.
I love that it's in your kitchen.
Yeah, I can quite assure you, my wife really does not like this in my kitchen. It's quite often on our kitchen table. She's away at the moment, so they can stay there for a few days, so it's good. And so as a relatively complicated surface unit, it can store video on it, or we can bring it to the surface. We can bring the video feed to the surface and record 4K raw footage as well. And that does create limitations as to how long the cable can be for the ROV, as you can't send that along a super long cable. I'll sit back down. And happy to answer any questions people have.
Any challenges with using that?
Many challenges. So our perfect situation is to have a boat that's either got some kind of a dynamic positioning system or be anchored. So the trouble is most smaller boats which we would typically deploy it from don't have dynamic positioning. They might have I guess some of the newer boats have a type of that. But most of the boats we tend to use anchor. And most skippers, in my experience, don't particularly love anchoring below, 60, 70, 80 meters. We do have a few people who are comfortable anchoring a bit deeper, but anything over 100 meters, it becomes quite difficult. It's also it's a challenge using it. There are often pots, lost fishing line on the bottom to get tangled in. Cable management is super important. So you've got, you might have 250 meters of cable out, not getting that wrapped around the prop. We've done all these things. We've had it caught in things. We've had it caught around the prop. We've all those things have been a problem through time. But yeah, you just manage it, I guess, the best you can. Currents as well. Oh, you've gone off, Barbara. I think you've pressed mute.
Sorry. If you have any questions, please come off mute. You're welcome to raise your hand or come off mute and ask your question. Are you using AI at all with your ROV or?
Yeah, so the ROV itself is human driven. So it is completely, it doesn't have any AI built into the ROV itself. So it's completely driven by us. For the work that we generally do, the kind of autonomous ROVs are not quite there yet. We tend to spend a lot of time very close to the reef, and a lot of the mapping work from some of the AUVs, at least the smaller ones. I'm not talking about NOAA's $10 million or $20 million ones. The ones in our kind of price range generally end up being a bit further away from the reef than what we do. So no, it doesn't. We use for a lot of our image analysis now. So we collect a lot of videos and a lot of pictures. A lot of our image analysis is increasingly being done by AI, much to the disgust of my students, because mostly I pay them to analyze all the videos and pictures, and now they're not super happy they're being replaced by a machine, but our funders typically are.
Go ahead, Tricia. Let me see, you're on mute. Yep.
Got it. The southern tip of the South Island, what sort of currents are there going around that tip? And what sort of temperatures are we talking about during their summer?
So our summer temperatures down there would probably reach, let me think, what would it be, about maybe 15 to 16 degrees. Currents can be extremely strong. You could see from that video as well, one of the big differences to that location compared to other places where we put the ROV is that there's very little sediment. So it's a place called Stewart Island. So it's actually a relatively big island off of New Zealand, which has been relatively untouched on land. So there's lots of natural forest. Hasn't been developed hardly at all. Not lots of rivers draining in there. So it's like the visibility was probably at that depth, maybe 40 meters. You could see the reef from like a really long way away. Yeah.
Wow. Thanks.
You're welcome.
Anything that surprised you about that zone that you're looking at that you didn't expect?
Yeah, I think just the amount of diversity. So there are pretty much in all of the times we put the ROV in, there are new species. You know, there are a bunch of species that that we haven't seen before. We don't know what they are. And I think also the diversity of the overall communities across New Zealand. I think people thought that we would find like the same thing pretty much everywhere we went. And that's not the case at all. We find quite different communities in all the different places. I still think that the sea squirts are the most unusual. There are elements that are shared between different places, but the sea squirts were just completely different to anything that we'd seen before.
When you
Go
said
ahead.
that, I'm sorry. When
No.
you said that 20 million sponges died off, you estimated that in what area? All around New Zealand?
So that was specifically in the kind of Fiordland area. So I had that and the map where there was the view kind of staring down. So it's an area. I'm trying to think what that coastal area would be. It'd be an area of maybe 100, maybe 150 kilometers of coastline. So yeah, so it was a substantial mortality event. It kind of also puts into context, you know, how, I mean, there was still lots left, and there was still, you know, there's 1,700 sponge species in New Zealand, and the majority of them weren't impacted. But there are these, you know, creatures in the ocean that there are, you know, millions and millions and millions of them, and you know, we know pretty much nothing about lots of them.
Do we know anything about their rate of reproduction on sponges? You say they're super ancient, 550 million years old. How long does it take for them to reproduce? Do you have any idea?
So, yeah, so the species, the group itself is that old. So some sponges are thought to live maybe as long as 10,000 years. Individuals have been suggested in Antarctica. Yeah, there are sponges in New Zealand that have been in New Zealand longer than people have, which is quite cool, I think. You know, which is, you know, 1,000 years or so. They generally are slow growing. They take a relatively long time in most cases to reach kind of sexual reproductive maturity. But they also they grow generally quite slowly. So if they get disturbed or if, you know, something wipes them all out, in most cases they take a long time to recover from what we know. It's hard to study things that take ages to recover, but I should say that's not all sponges. Some sponges actually have the opposite. They grow very, very fast. They don't live very long, and then they, you know, die quite quickly. So they have that range. But yeah, some of those glass sponges I was showing you, they can be 10,000 years old.
As a follow-up to that, when we've had warmer waters here, we've seen what we call urchin barren areas. Sunflower starfish have died off or gotten diseased, which kept the urchins in check and, you know, destroyed the kelp. Are you seeing those kinds of shifts, on top of which we also see warm water tropical fish coming further north because there's dead zones in Mexico and fish coming down from Northern California because they follow the food.
Yeah, that's right. Yeah. We have urchin barrens in New Zealand, but driven by overfishing largely. So the snapper that I showed you in the video, and also crayfish are major consumers of urchins, and the loss of those snapper and those crayfish have caused urchin barrens in lots of areas, particularly in northern New Zealand, but pretty much all the way around New Zealand. I guess that's really well known from studies that people have done in marine reserves. So areas where have been put as marine reserves, and the snapper come back and the lobsters come back, and then all of a sudden the urchin barrens disappear, and the kelp forest comes back. In those deeper environments, we don't tend to see so many urchins. There are not that many larger invertebrates kicking around. They all tend to be kind of smaller stuff. Yeah.
Go ahead and come off mute if you'd like to ask a question. So you've been to Wakatobi and a lot of other environments. I'm taking, how do you think the New Zealand reefs at those depths compare?
Yeah, so I have taken one of our ROVs to Wakatobi, and in the depths in the Wakatobi, they are also completely covered in sponges. So it's a very spongy environment down below, kind of 60 or 70 meters on those reefs too. And you find kind of huge barrel sponges. The picture I showed you of me next to a barrel sponge at the very beginning, that was actually taken at deeper than I'm allowed to dive to. Oh, you're recording this, I shouldn't tell you this, but it was a long time ago. Deeper than I'm allowed to dive to with the university, to actually recover the ROV because he got stuck on something. And just while I was down there, we saw this huge barrel sponge, so I couldn't resist getting a picture next to it. But yeah, those deeper water environments are very much dominated by sponges, kind of similar to New Zealand, but different species, obviously, and bigger.
Yes, more questions. You know, I was kind of amazed about the amount of water flow through a sponge, you know, the pumping action of that. You know, it seems like if we could harness that and multiply it, that would do a
Yeah.
lot to,
Yeah, I've had students before who've talked about the potential for giant barrel sponges to stick a little mini turbine over the top and harness the energy from them. But yeah, they are amazing, and it's worth next time you're diving along is thinking about the activity that they're doing. They're having that really big impact on the turnover of water in those environments. Yeah.
I'm sorry if I missed it. How did you get into this specialty? Sorry, sorry if I missed it earlier. How did you get into this area of interest?
That's a good question. I guess my title gives that away a little bit maybe of my talk. So I've obviously always been a diver, and I've worked in a few places where there are these kind of reefs, but in shallow water environments. And I guess I said that our ROV is called George. It's named after George Mason, who is now passed away. He visited our university and was very, I guess, one of those chance encounters. He visited our university and was looking for projects to sponsor and fund. And I said, you know what, I'd really like to know what it's like below the depths of which I can go diving. And he's like, well, how could you do that? And I was like, well, you could buy me one of these new fancy ROVs. And that's kind of where it started. So it was an interest driven by curiosity, like I guess a lot of science is. Yeah. So and I guess things went from there. We started with a cheap ROV, and we've progressively gone to more and more expensive ROVs, which give us, I guess, more capability and better video ability and ability to put sensors in and take sensors up.
Excellent.
But
Thank you very much.
it's kind of one of those areas. Yeah, it's kind of one of those areas that's kind of exploding in terms of interest. There are lots of people. Our group has been some of the first to highlight these communities, and people are starting to work on them all over the world.
I guess I wonder, go ahead, Tricia.
I was just wondering what's the attitude of the New Zealand government towards overfishing? Are they quite strict, or are they quite lax, or somewhere in between?
I guess it depends. We have, I guess, I consider relatively short government cycles. We have our government at the moment that is quite keen on fishing, economic game. Whereas if we have a more, I guess, left type government, we tend to have a more conservation, sustainable management focus. Our fisheries are generally ranked as one of the most sustainably managed in the world.
Right.
If you ask our conservation agencies, they would disagree. But overall, most of our fisheries are considered to be quite good. They're obviously harvested a lot. There's a lot of impacts. There are a lot of things that aren't great, but I think compared to many places in the world, they're held up as an example of how fisheries can be sustainably managed. I don't want to give an impression that it's all fantastic and great, because it isn't, and there are certain fisheries that are worse than others. But yeah, the government recognises that if you take all the fish today, there won't be any fish for tomorrow. So the long-term sustainability of that industry is dependent on there being more fish and you looking after the resources.
Are they more protective of the South Island?
No, it's equal. I guess New Zealand is small. We only have a small population. The government focus is generally equally managed, and the fisheries are managed as kind of a whole. So so there's a central organisation that manages the fisheries, and yeah.
Thanks.
That's okay.
So you've written a lot of papers, you've done a lot of research. I'm wondering what keeps you inspired.
No, that's a really good question. I actually really like making new discoveries and going new places. Many of my colleagues at my age and stage of career start not leaving the office, and they spend all of their time in front of their computer. That's not me at all. I think if I couldn't go into the field still and go diving and go out with the ROV and go on boats, then I probably would stop very quickly doing what I do. I work in a university. I really enjoy teaching. I think teaching is super important. I get a lot of joy out of passing on what I do, working with graduate students, doing research with the students who are out in the field with us all the time. But also it's equally fun trying to inspire, first year students to want to stay on in marine biology, especially when they look around and see, all they see on TV is doom and gloom and how terrible things are. And not everywhere is like that, not everywhere is doom and gloom.
So it sounds like the reefs around New Zealand, even though you've had this massive loss of sponges, or would you say they're fairly good shape, health?
Yeah, I would say so. We've got I don't know, I want to say 40 plus marine reserves, which cover about 10 to 15% of our marine environment, which are full take, no fishing, no nothing marine reserves. We would like it more than that. There are also a whole bunch of other types of protection in place, seafloor protection areas and things like that. So, most people, I guess, like me, would like to see more. And I guess most people who are recreational or commercial fishers would like to see less. And it's, I guess, balancing those different viewpoints. I think New Zealand does a generally good job of doing that. I travel a lot. I see a lot of different places, and I see the things that we do here generally being quite good compared to lots of countries that I visit. Wakatobi is a good example. Indonesia, has terrible plastic problems. The plastic in the marine environment is unbelievable in some places. In New Zealand, that's not a problem. We're so far that fortunately most of it doesn't seem to float here, and we manage it quite well locally. So we don't have those problems so much.
Go ahead.
What's a good way to get involved if someone's interested in underwater drones and vehicles from a recreational standpoint? I was a oceanography major 25 years ago, and it required a complete vessel to do what a small drone can do now. I never worked in that field, but I would like to kind of start to explore it again. So what are some resources and recommendation that you could recommend, and where do you see the ROV industry going?
Yeah, to answer the first question, that's a really good question. I think so. There is every entry level to high-end drones now, which in my experience produce really good results. So we have, as well as having this expensive drone, we also have a much cheaper Chinese drone, Chasing. It's called is the brand, and they make drones from kind of $10,000 down to maybe $1,000. So in the realm that a normal person can go out and explore those things. And then while kind of the cameras in those might not be that great, most of them give you the ability to strap on a GoPro or a DJI Osmo, which means all of a sudden you've actually got really quite fantastic footage for the fraction of a cost of what my ROV is. I think sadly you asked about where things will develop. Sadly, I think it will get to the point where I won't need to go in the field with my ROV. I'll probably just chuck it in Wellington and it'll go off on its own program thing and do its own thing, which would be very sad for me. But I suspect that in countries where or areas where there's a lot of interest, or places like marine reserves where they want to do ongoing monitoring, then that's what will be happening. It won't even need to come out of the water, except to be cleaned. It'll go in its little docking station. And I've seen those. Those drones are available. They're double or treble the price of what mine costs. Or maybe even more than that, and they're usually bigger, but they will get smaller, I suspect, and they'll get more able to do things on their own. And not need a cable. So obviously ours is cabled, which creates all sorts of limitations. But it does also mean we can see what it's doing when it's down there.
What do you think, Barbara? Maybe a drone class from Malibu Divers at some point?
I'm taking donations.
Absolutely.
You can name it. We'll take your first name and your last name. I mean, I'm all into it, I think it's cool, but I never want to give up diving, be it scuba or free diving, so.
This is it, you can't. I guess not without specialist training.
Yeah,
Dr.
we have one for a member of the LA County Sheriff's Dive Reserve Team, and we have one, and you know how often we use it?
0.
Yeah,
Yeah,
there's
so
very few
we-
people who've been trained on it. It's very big, it's very heavy. It's an
Yeah.
older generation. I think the thing must weigh like 500, 600 pounds, you know?
Yeah,
So,
a number of research organisations around New Zealand have brought them to kind of replace divers, but actually, you know, it's still much easier to put divers in the water for research work or for monitoring work than it is to use the ROV. Yeah, it's much more straightforward.
now, the regular sworn sheriff's team has the really fancy one, and they use that quite a bit. So Homeland Security, missing people, missing cars, whatever it is, planes, you know,
Yeah.
so, but it's very hard. People have no idea how hard it is to drive it, maintain it, you know, the whole bit.
Yeah, that's why mine's on the kitchen table, because it does take a lot of, it's always in pieces, being, you know, greased, checked, cleaned, make sure it works so it's ready to go. Yeah.
Very cool. What other questions do people have? Did you mention, is there a way for people to get involved with research around this, or?
I guess it's true. It's tricky at the moment. There aren't specific ways that that people can help us. We're always looking for new locations to go to. But yeah, so I guess because it's specialised. But you know, if you're in New Zealand, then you know, come and pay us a visit.
There you go. So I think the water environment is very similar. We don't have the corals, except very,
Yeah.
very deep with the dark blue or purple hydrocorals, but temperature-wise, it seems pretty similar.
Yeah.
Dr. Bell,
Yep.
in the future, do you anticipate doing research going beyond the Mesophotic zone?
Yeah, that's a good question. So yes, I guess is the answer to that. Yes and no. It depends. There's another zone that's slightly beyond the Mesophotic where the light stops. But the problem is that we're not problem, but lots of people have kind of more worked in those environments. So once you get below kind of 200 and 250 meters, it's kind of considered the deep sea, and that's where all the kind of deep sea biologists work. So I'm most interested in that bit where there's still light, and there's a kind of transition zone between the shallows and the deep. So yeah, maybe the deep sea's cool, but there's already lots of people working in that space. So I guess I found a niche to do something slightly different. That's a good question though. I do. So the ROV is constrained in terms of depth, firstly by the dome, and secondly by the cable or the feed from the camera to the cable. There are some upgrades it can have so that the high resolution camera feed can transmit up the cable and still be seen at depths greater than 300 meters. But I haven't gone down that route yet. So then it could get to kind of 800 meters, which would be kind of cool. But then typically you need 3 times the cable as it is deep. So you know, yeah, you need like a ridiculous. I think you need a bigger boat and stuff. Yeah.
Thank you.
Cool. So we're coming up to the end of the hour. Really appreciate it, Dr. Bell. And if you have any other questions, please send them to me. I'll send them on. Oh, somebody's just coming in. They must have the wrong time zone. So yeah, it's all good.
All right. Thanks everybody for joining. It's great to be able to share what we do.
Thank you, Dr. Bell.
Thank you.
Thank you.
Thank you,
Nice to
Doctor.
meet you all.
Thank you, Barbara.
Sure.